sene sonra geldi.
Merhaba hocam.
Sen olay olmadıktan sonra Türkiye'ye döndün.
Evet. İyi olmuş aslında benim oraya gitmediğim.
Evet.
Yoksa ortada kalırdım yani. Evet. Ne oldu, o tenür mü alamadı, ne oldu, niye döndü bilmiyorum.
Ailevi sorunlar yüzünden döndüğünü
öyle mi? Ha.
Bilmiyorum ya.
Doğrudur.
Merhaba.
Abi sağdan mı geliyorsunuz? Aa, ne kadar güzel. İnterneti bekliyoruz da oradan yayın yapılacakmış. 1-2 dakika içinde başlarız herhalde. Söylerim. Senin isim neydi abi? Emrecan. Soy isim? Söylemez. Tamam.
Hendesehaneydi ilk adım. Aynı zamanda Osmanlı İmparatorluğu'nun bilime, tekniğe, mühendisliğe attığı ilk adımdım. Yıllar içinde adım ve binalarım hep değişti ama yönüm, vizyonum hiç değişmedi. Sınır tanımayan mühendisler yetiştirdim kurulduğum günden beri. Talebelerim gemi mühendisliğinden harita mühendisliğine her alanda yeteneklerimizi ülkemizin hizmetine sunarken ben bünyeme yeni bölümler, teknikler kattım. Osmanlı İmparatorluğu'nda, askeri teknolojide de, bina ve yol imarında da, Hicaz'a uzanan demir yollarının arkasında da yetiştirdiğim mühendislerin tekniği, çalışkanlığı, vizyonu ve emeği vardı. Kurulduğum ilk yıllarda da hayallerim hep yerden göğe kadardı. Mizah zekanın zekatıdır dedim. Şaka dergisinde espriye, karikatüre, bilim ve teknik kattım. Öğrencilerim 1909-1928 yılları arasında mizahın mühendisçesini yazarken dünyaya o günlerde de başka baktım. Söz uçar, yazı kalır sözünü kendime şiar edindim. Kurulduğum günden beri yayın faaliyetlerine ayrı bir önem verdim. Ülkemizin faaliyeti bugüne kadar devam eden...
Evet, merhaba değerli katılımcılar, hepiniz hoş geldiniz. Bugün yine gurur duyduğumuz bir mezunumuzla birlikteyiz. Mezunumuz bize, özür dilerim adını söyleyeyim önce, Utku Sönmez. Şu anda Carnegie Mellon Üniversitesi'nde gözüküyor. Mikro akışkanlarla ilgili bir konuşma yapacak, seminer verecek. Kendisi oldukça başarılı. Birçok yerden aldığı teklifleri değerlendirip şu anda Levent Trabzon hocamızı davet ediyorum.
Teşekkür ederim hocam. Ben de Utku öğrencimize teşekkür ederim. Kendisi 2016 yılında lisans mezunumuz. Bitirmeyi beraber yapmıştık ve çok başarılı bir bitirme gerçekleştirmişti. Bitirme sonucunda bir SC yayını, bir Amerika patenti ve aynı zamanda başka ödüllerde, yarışmalarda ödüller kazandı. Daha sonra Carnegie Mellon University, daha sonra orada doktorasını tamamladıktan sonra geçen sene 2021 yılındaki tıp alanında Nobel ödülünü kazanan Ardem hocayla önümüzdeki 2 yıl içerisinde postdoctoral'ını yapacak ve bugün de doktora çalışmasını yaptığı mechanobiology konusundaki çalışmalarını bizimle paylaşacak. Teşekkür ediyoruz katılım için tekrar.
Çok teşekkürler geldiğiniz için. Salonda olanlar ve internet üzerinden takip edenler. Ve introduction için de çok teşekkür ederim. Konuşmamı İngilizce yapacağım. Aramızda yabancılar da olduğu için kusura bakmayın bunun için. The name of my presentation is going to be studying the mechanically regulated cellular behaviors by microfluidics. I'm going to start with a quick introduction of microfluidics in biomedical sciences as well as mechanobiology. And then I will be talking about 4 research projects that I led during my PhD at Carnegie Mellon University, and I will talk more in detail about those. But first, let's define what microfluidics is. It's the science and engineering of systems that can process very small amounts of liquids and gas, so fluids, in microfluidic channels, which has the ranges, which has the diameters or dimensions that are ranging between tens and hundreds of micrometers. Basically, the thickness of hair is what defines the dimensions of these systems. And if you look how this technology has been developed, we see a critical breakthrough in 1955 with the development of silicon-based microfabrication technology, which is the technology that now enables us to fabricate semiconductor chips. And we use the same technology to manufacture microfluidic devices. And in 1998, George Whiteside's group developed a fabrication technique called soft lithography, which enabled these devices to be fabricated in any ordinary lab. Before that, you really needed state-of-art clean rooms, which you cannot find in all the universities. But after 1998, everybody was able to create these microfluidic devices. And if you look at the literature, at 1998, there is this trend that right now skyrockets, and everybody can make, design, and use microfluidic systems for their research projects. And right now, there are more than 5,000 projects published every year in the literature. And slowly but firmly, these devices are making their way to market. And by 2026, it's expected to surpass $20 billion market size, which is astonishing. And if you look at the biomedical applications of microfluidics, which is what I'm interested in, there are lots of them, ranging from diagnostics, organs on a chip, personalized medicine, genomic studies, 3D bioprinting, and drug discovery. I can talk hours about what they are, but now I'm going to be focusing on different parts of it. And if we look how we, in a nutshell, fabricate these microstructures, as I said, we borrowed them from the semiconductor industry. We get a silicon wafer, a semiconductor substrate, and we coated that with spin coating with photoresist. And then we expose this photoresist selectively using a mask to UV light, which hardens the photoresist, and we remove the non-hardened part of the photoresist, and we obtain a mold. We process that mold, and then we use a polymer called PDMS, polydimethyl siloxane, which is in a liquid form, and if you mix it with a curing agent, over time, with the help of the heat, you can cross-link it and make it solidify. So we pour it on top of the mold, and then we peel it out once it's cured, so we replicate the surface topology, the microfeatures on it. And then we permanently bind it to glass slide to obtain the microfluidic channels that are as complex as it can get. And I started doing microfluidics work, as Dr. Trabzon said, when I was an undergrad here in the İTÜ Mechanical Engineering department, and we were working on the cell separation and enrichment in microfluidic devices using a microfluidic channel configuration that we have developed. And people like that approach, and we published our study in an international peer-reviewed journal. We received United States patent for that. I presented the results in an international conference in United States. We also got the German patent too. So it was a good start, but this is not something unexpected or unusual in this department, because if we look at the timeline of microfluidics, I would like you to focus on the first mention of microfluidics in 1964. Guess where it came from? It came from Cemal Erringem, which is a graduate of our university, of our department, Mechanical and Aeronautical Engineering at that time, from which he graduated in 1943. And he enabled, basically, or he conceived the idea of microfluids, which led to $20 billion+ market size right now. On the other hand, one of my other interests, and I combine this with microfluidics, is mechanobiology. I'm a mechanical engineer who is interested in biology, so it's not a surprise for me to go there. And mechanobiology is basically biomechanics, but at the cellular level. And when you go to the cellular level, you see that mechanics actually affects the biological processes at a very high rate. And there are studies that show that mechanics contribute to the development, to the differentiation, the immune response, physiological and pathophysiological cases. And if we go down to the cellular size, there is no clear distinction between other considerations and mechanical considerations. Therefore, this field is this field is inherently interdisciplinary, because you have to take biochemical concerns. You have to think about biophysics and molecular cell biology and genetics. So people come together generally to run these collaborative projects. And microfluidics have been used in these research. People made these substrates on which cells can be selectively adhered, and they show that cell fate, their apoptic states, can be controlled by the topography of the substrate that are microfabricated. This is super interesting. They get stem cells and seeded on a hard substrate. I shouldn't say hard, substrate with a high Young's modulus, so it's more rigid. And these stem cells turn into bone cells by just changing the substrate's Young's modulus. And if you see those cells, the same stem cells, on a soft substrate with low Young's modulus, they turn into neural cells, because brain is more mushy and bone is harder. So they can sense their environment, and their differentiation can be controlled by substrate. People can use these micropillar arrays to measure the forces created by cardiomyocytes, the muscle cells in heart. And they can create these cell stretching devices to mimic the blood flow and the radial expansion that is caused by the pulsatile blood flow on endothelial cells. So there are a lot of interesting things that you can do with microfluidics in the field of mechanobiology. And I have done 4 of them during my PhD, and I will talk about those now more in detail. The first project is about the mechanostimulation of fruit fly embryos to see how mechanics regulate the development. And now let me show what I have done in this. So we use fruit flies as model organism. You may think that fruit fly, what the fuck is going on? Like they have nothing to do with the real human. But we share 50% of our genes with fruit flies, and fruit flies are actually the model organism that taught us how genetics works. It led to 11 Nobel Prizes. People got Nobel Prizes, 11 of them, by just using fruit flies. And the way that they develop from embryo to adulthood actually, from biological point of view, replicates quite accurately what we experience. And during this developmental process, there is a really interesting stage that's called gastrulation. In this gastrulation stage, as you can see in the video, the same epithelial cells are migrating towards each other, and almost like in a buckling fashion, they are compressing each other, and then they buckle inwards, creating this structure called mesoderm invagination. And this is a very critical process because it marks the state where the 3 germ layers are formed, which then defines where the organs and the tissues are going to be developing inside the body. That's why Lewis Wolpert said that the most important the time of your life is not your birth, marriage, or death, but gastrulation. If the animal cannot go through gastrulation properly, it dies. There is no more development. There is no reaching to adulthood. And this process, as you can see, is super mechanically active. They are moving, changing shapes, and then bending and buckling, and it is controlled by genes. But the question is, if there is a feedback loop, if mechanics can control the genes, which is a big question. And we know how genes control mechanics. It's basically, let me move the slide. There are basically some transcription factors. These transcription factors are the proteins that defines which genes are going to be activated. And one of them is Twist, which is the main player here, that controls this gene activation, which leads to the mechanical activity. And people, in order to see the feedback loop, what they did is they applied compression externally to these embryos to see if they can change the gene expression. And they were using really primitive stuff. They were just getting 2 plates of glass, and they were putting the embryos in between, and they were compressing them like that without knowing how much they compress, how much force they apply, if they're killing or not, or if they apply compression or not. So what we thought is, if we can make a system that can precisely apply mechanical compression to fruit fly embryos to see which proteins and genes are being activated at the end of this mechanical stimulation, and we want to do it for hundreds of fruit fly embryos so we can get statistically significant results. And this is the microfluidic system that I came up with hypothetically. So there is a center channel where we put the embryos. The center channel has 2 side channels on each side, which are gas channels, and they are dead ends and pneumatically activated. And in between, there is a deformable sidewall made out of PDMS, which can be deflected inwards if you apply positive pressure and outwards if you apply vacuum. So in order to, this is the top view, by the way. This blue is the embryo inlet. The embryos are accumulated here. On each side, we have gas chambers, and we apply pressure to deform these thin deformable sidewalls. And I created this analytical model to see how much deflection we can apply to these walls with the given pressure. And I made some simple numerical simulations to see if they are fitting each other. And after that, I get a rough estimation of what the dimensions should be. And I fabricated this proof of concept models on silicon wafers, and I made PDMS devices out of them, and I put embryos in it. These embryos are really small. They are around the thickness of the hair. And by changing the pressure, as you can see, I can compress them, which work really well. And I got the experimental data as well. And it looks like the theory, experiment, and the simulation, they all fit each other. So I finished the characterization with this graph where I show the PSI versus embryo compression. The next step is to make it high throughput. And it is easy to make. You just make it longer and then parallelize it. Now you have hundreds of embryos that can be compressed in the same way. And this is the final device. So we have this narrowing atrium where the embryos are automatically aligned. And I have a bottleneck at the end where the embryos stop because they cannot squeeze through this little channel. And I can easily load these embryos inside these channels by just tilting the microfluidic channel after introducing the fruit fly embryos to its inlet. And by just tilting a little bit, in a couple of minutes, I can introduce hundreds of embryos. And under vacuum, they are moving freely. If I remove the vacuum, they are immobilized. And if I apply positive pressure, the walls deflect inwards and compress the embryos. So these are the results that we got. We want to measure the twist expression as a result of the mechanical compression. And as you can see, these are the hours passed after compression. And these are the amount of compression that we apply. And it looks like if we apply mild compression for a long time, we can really induce the expression of twist, which is almost none at the beginning, and it is extremely expressed in an ectopic fashion, meaning that homogeneously everywhere because we apply bulk compression, unlike the local compression that is happening during gas relation. And if you look at quantitatively, we can also see that there is a dose and time-dependent mechanical response coming from this twist expression in this device. And if we apply really high, 22% strain on fruit fly embryos for a long time, the twist expression is not high anymore because we kill the embryos. And you can see it from here. So in the control case, 75% of the embryos are making their way to adulthood. All right? This is pretty classic. These are the things that people have known for years. But if you apply a mild compression for a long time, or if you apply high compression for a little time, what you see here is the viability of the embryos are skyrocketing almost to 100%. So it's almost like the embryos cannot create enough compression by themselves, therefore they cannot increase the twist, which regulates gas relation, and they cannot go through gas relation, and they die, like 25% of them. With a little push, by applying a little bit compression, we can make these embryos more healthy, I will say, healthier. And I made an illustration depicting this, and the referees and the editors really liked this study, and they published it at the cover of this Royal Society of Chemistry journal called Lab on a Chip. So in summary, I made a mechanical stimulation device for fruit fly embryos. I characterized this analytically, computationally, and experimentally. And I show that twist expression is dependent upon compression in a dose and magnitude depending way, in short. So this is the first project, pretty much. The second project is about how endothelial cells that makes our blood vessels, are affected from blood flow. In other words, affected by the shear stress that is created by the blood flow. And we specifically studied an interesting rare disease, which is called HHT, hemorrhagic telangiectasia. And this is hereditary, meaning that it's genetic. And what happened is, these are arteries, these are veins, and they are connected to each other by capillary. In this disease, the capillaries are expanding, and they create this vascular shunts, which leads to internal bleeding. And we know really well genetically why it happens. It happens because of a mutation in a gene. But physically, what leads those capillaries to expand, it is unknown. But we had a hypothesis. So what we claimed is, under normal circumstances, the endothelial cells that are at the tip of the arteries are migrating backwards through the arteries, which leads to have lesser cells in the capillaries, which makes them small. But when there is a mutation on these ALK1 and endoglin genes, then endothelial cells cannot sense the blood flow. They can't go against it, and they are being dragged by the shear stress towards the capillaries, which increases the number of cells in the capillaries and makes them expand. So we want to test this, and I have to specify a specific shear stress pattern in this artery to capillary junctions, where we have shear stress gradients. Here the shear stress is high, here it's low, and there is a negative shear stress gradient from artery to capillary. And we know how migrating cells looks like under microscope. what happens is these cells are polarized in the way that they migrate, that they move. Their nucleus remains at the trailing edge, and their Golgi apparatus are at the leading edge, and almost like they are facing towards the direction that they are moving. So by looking at the cell polarization, we can see where they are going actually. So I want to create a microfluidic system that can create these different levels of shear stress and shear stress gradients to see if this hypothesis is true or not. And making this uniform shear stress channels are easy. Just make a rectangular channel and you can create the shear stress that you want and you can compute it analytically. But having the shear stress gradients are a little bit tricky because we want to have a linear shear stress gradient so that all the cells are exposed to the same gradient slope. And you can have it by following the wall shear, the wall equation of the side walls, which depicts a state where the shear stress becomes linear. And I came up with this formula, and I built this device. So let me put it down in parts so it will be clear how it works. So there are these uniform shear stress regions with different shear stress magnitudes. Here the shear stress is high because the area is low. Here the shear stress is low, but they are all uniform. We have exactly the same regions on the other side just for internal control. And here in this connecting regions, we have shear stress, linear shear stress gradients. And if the channels are expanding, then the shear stress gradient is negative because shear stress is decreasing. But if we narrow the microfluidic channels, then they have positive shear stress gradient. And we have the same shear stress gradient with a different average shear stress magnitude. So we can decouple shear stress from shear stress gradient. Because here, the shear stress gradient is the same, but the average shear stress magnitude is different. So I made some simulations on this device. I experimentally measured the flow rate and the flow profile, and I then seeded the cells inside it. I created an endothelial cell monolayer. I built a stereo flow circuit because cells cannot live in a contaminated environment, so everything here is autoclaved and they are disposable. And it is compatible to microscope, and I can see the cells under microscope. And I made a computer vision software which can find the nucleus of the cell and the Golgi of the same cell. They pair it and they see which way the cell is facing. So it can detect automatically the polarization of the cell and it color codes it. So if the cell is facing upstream as if it is migrating against the flow, it is red. If it is facing downstream of the flow, it's blue as if it is going to the direction of the blood flow. And these are the polarization results that I got. If we look at the non-flow case, the static case, half of the cells are not polarized, they're staying there, and the rest of them are migrating in random directions. I created 4 bins and they're all the same, left, right, up, and down. But if I apply shear stress, they polarize against the flow as if they are moving against the flow in a dose-dependent way. As the shear stress increases, they are polarized more against the flow. And if I show you the shear stress gradient regions, again, no flow, no directional polarization. But if you look at the case where I showed you the regions where the average shear stress magnitude is the same, but the shear stress gradient sign is the opposite. So here, negative shear stress gradient, here, positive shear stress gradient, average shear stress is the same. So it shows shear stress magnitude independent polarization of against flow, which is favored when it is negative, just like the case where we have this artery to capillary junctions. So cells are more tend to move against the flow if there is a negative shear stress gradient. And regardless of what kind of a shear stress gradient or what kind of an average shear stress magnitude you have, you see the same pattern. Left is always more polarized against the flow. It's more red than the blue one. And I changed the dimensions of the microfluidic channel. I made exactly, I followed the same design guidelines, but I made a microfluidic channel that has totally different dimensions, and I applied different shear stress and shear stress gradients, and the results were exactly the same, which is kind of also a control. And I have also seen some features, I don't want to go much in detail, that are associated with cell quiescence, meaning that cells do not proliferate when they are exposed to shear stress, regardless of what kind of shear stress magnitude or gradient you have. So the polarization seems to be the only case that can explain the accumulation of the endothelial cells, because they could have been polarized and also proliferate, and you don't know if the capillaries are expanding because of the proliferation or not. And we submitted and published this work in Lab on a Chip. This time it made it to the inside cover. So in short, I made a microfluidic device that can enable me to create 14 shear stress modalities in the same device. So I can run 14 experiments in a single run, basically, which makes things really easy. I characterize this device again analytically, computationally, and experimentally. I show that the polarization of the cell, which is Golgi nucleus orientation, is proportional to the magnitude of wall shear stress. And while the positive wall shear stress gradient inhibits, negative shear stress gradient enhances the polarization of the cells against the flow. Of course, this is just the beginning, and people are still using in our group these devices, and they do live imaging over time to see how the cells are actually moving to further work on this hypothesis, because what we want is to see if they are moving against the flow or not. And we are also creating endothelial cells that has this mutation intentionally to see if the migration patterns are changing or not. The third project that I will be talking about is actually exactly the opposite of what I have told you. So we are going to look at the motility of immune cells, our defense mechanism, and how they move in our body. And these cells are, unlike the endothelial cells, are not attached to the substrate. So if we apply shear stress, they are being dragged by the flow. That's why, that's how they are transferred inside the bloodstream. But inside the tissue, they create their own locomotion to move inside the solid tissue. And that's what we are interested in. So we want to create a system that can exclude all the forces that are fluidically being applied to the immune cells, which would enable us to see how they would move without the drag of the flow, how much motility that they can create. And this is very relevant because unlike endothelial cells, immune cells are constantly motile in our body. Let's say you have an infection in your hand. The infection tells the immune cells to come here and deal with the infection. So they are migrating all around our body through the blood vessel. They get out and they find the exact location in the tissue where I have the infection, all right? Or if I have a bruise or anything like that. So the motility is essential. But how does inflammation, this infection, tell immune cells to come here, this specific location? It is being done by gradients of soluble molecules called chemokines. Once these chemokines are being secreted in the inflammation side by the injured cells, it spreads in our body and creates this gradient because they spread by diffusion. And the immune cells are following these gradients. They go towards the chemokine, and at the end, when the chemokine gradient is completed, then you have this inflammation side. And the same thing can work against the tumors as well. So this red one is an immune cell, a CD8 positive T cell, and the blue one is a tumor cell. And it attaches to this tumor cell after following chemokines, and it gives it a dead kiss, which leads to the activation of apoptotic pathways, and immune cells basically forces tumor cell to commit the suicide. But this only works well in the dish. In real life, our immune system is very weak against tumors. That's why we need some external intervention like surgery or chemotherapy or other things to kill the tumors in our body. And a recent approach to do that is immunotherapy of cancer. Why we cannot make these immune cells more stronger so we wouldn't get any external drug in our body, but we will make the immune cells fight the cancer cells in a more effective manner. So, since we were interested in the migration of the immune cells, I had to create a flow-free concentration gradient chamber in which I can create the chemokine gradients without applying any flow to immune cells so they can move by themselves. And this is the design that I came up with. So it is 3 layers. In the top layer, I have 2 flow channels. And I let the flow go through the first microchannel, which is just like 200 micrometer of the width. And there is this semi-permeable membrane, the gray one, that stops the bulk flow, but it lets the diffusion. And this chemokine solution diffuses into the gradient chamber, where I will have the immune cells. And if I have a sink channel, which is again flowing, the pressure balances, and the 2 sides diffuse to each other, and I have a linear gradient of the chemokines. So this is the simulation that I made to show that. And I made like different variations of the chambers to see how the aspect ratio and the geometries of the microfluidic channels are affecting the gradient generation computationally. And I came up with like a reliable microfluidic channel dimension which can be microfabricated and can create the gradients that I wanted. And this is kind of how it looks like. There are like 2 gradient chambers, one on the left side and the other one on the right side. They create the same gradients, but the steepness of gradient is different because one of them is wider, where you have a shallower gradient, and the other one is narrower, where you have a more steep gradient, but the same linear gradient, to see if the steepness of gradient affect the motility. And there's a cell introduction port in the middle, by using which we can put the cells in it. This chamber is like 0.5 millimeter. But it has 500-600 cells, which I can track directly by using a software. And I created these, this chemokine gradients experimentally as well, and I showed that this works fine. And I then put the cells in it. I used an immortalized T-cell line called Jurkat cells, just for proof of concept purposes. And I can track them under microscope. You cannot see it well here, but I will show you other videos as well. And these cells can only move if they are placed on an extracellular matrix, the proteins that makes the architecture of our tissues. They attach these proteins that are basically on the surface, and as we increase the concentration of this extracellular matrix called fibronectin, we see that cells are moving faster and faster, which is expected. But if we create the gradient, which I have done next, I have seen something interesting. If I look at the same chamber, and if I split the chamber into 2, the gradient chamber, if I look at only the right-hand side, I see this gradient, and the cells are following the chemokines, which is to the left. But if I look at the left-hand side of the gradient chamber, where there's the same gradient, but the average concentration of the chemokine is high, they move randomly. So almost it's like the cells are being saturated by the chemokine, and no matter if there's a gradient or not, if all the receptors are full with the chemokine, then they cannot sense which direction to go. And here, almost like 50 nanomolar concentration marks the saturation. And we can see that also the quantitatively, the chemotactic index is totally different between 2 cases. And if we compare the steep and the shallow gradients, we see the same motility behavior, but with one difference. When the gradient is shallow, the cells are moving faster, interestingly. Their average speed is statistically different. And if we put all these things together, then we see some interesting pattern that can be physiologically relevant, even at this very basic level. So when there is an inflammation, chemokines are diffused in 3D, which creates this parabolic profile. And in the regions that are far away, that are distant from the inflammation site, the chemokine gradient is very shallow. And this is where the immune cells are. So they are fastly recruited to the inflammation site because the chemokine gradient is shallow. And they enter the inflammation site, And in the inflammation side, there's a gain gradient, but the average chemokine concentration is high, so they move randomly. This is super efficient way of looking for the cells, the pathogens, the virus, bacteria, whatever it is that causes the infection. And whenever they find, they just engulf it, they kill it, and they can keep doing it till the chemokine secretion stops. And if they happen to go out the inflammation side during this random motion, the chemokine concentration decreases and they are attracted back to the chemokine source. Next, we use murine, the T cells that we get from mouse. These are primary cells, and we want to investigate how these cells are moving in microenvironment that are associated with tumor, because we know that they cannot be effective, they cannot find the tumor cells inside the body. There are two considerations here. Tumors soak all the nutrients, in this case glucose, from the surrounding area, so these cells should function in an environment where there is no nutrition, there is no glucose. We call it hypoglycemic condition. Second, since there is no glucose, their mitochondria cannot work, the powerhouse of the cell. And it leads to mitochondrial dysfunction, which ends up T cells having exhaustion. So what we did is we targeted the mitochondria of T cell with a toxic substance that can be activated by light. So whenever we shine the light on the T cells, this toxic substance basically degrades the mitochondria without killing the cells. So it can simulate the mitochondrial dysfunction. So then we run these experiments, similar ones, under chemokine gradients and during the random motion. This one is from random motion. What happens is if we decrease the glucose concentration of the media, cells have the same migration parameters, but they are moving slowly. The only difference is here, average velocity. If we create a chemokine gradient, and if we compare the control cells with the cells that are in glucose media, we see that cells are slow again, but their persistence in chemotactic index as well decreasing. So they cannot follow the chemokine as well as the control cells. The control cells can go to the left where the chemokine gradient is more effectively. So the tumor microenvironment can imply that cells cannot follow the chemokine gradients as well, as good as the control one. Next, we look at the cells which has the mitochondria damage. And we have seen something similar where the average velocity is decreasing and the others are the same. The persistent chemotactic index is 0 because there is no gradient here. This is just random migration. And if we subject these cells, the same cells, to chemokine gradient, what we see is, again, they are slower, just like what we have seen in hypoglycemic environment, but their chemotactic index and persistence are increasing, meaning that they can follow the chemokine gradients better. So they are slow, but they can follow the chemokine gradients better. And if we do both of them, if we put the cell that has mitochondria damage in a hypoglycemic environment, we see exactly the same thing. The average velocity further decreases because both negative things are together, but the cells that has mitochondria damage has better persistence and chemotactic index. So there's a trade-off between these two, and some of them can be helpful, the other one can be more dangerous, but both of them slows down the cells. So in summary, in this project, I made a microfluidic device that can create this flow-free concentration gradients. The Jurkat cells created a pattern that is associated by the inflammatory response of T cells entering to the inflammation site, random migration going out and coming in back. And the T cells have shown that the mitochondrial dysfunction strengthens the chemotactic response while the hypoglycemic environment slows it down. This is in the nutshell what we have found, and this project also keeps continuing. And the last one is more like a short technical note that it has nothing to do with biology at this point. It's about a microfabrication technique that I devised, and it is called polycarbonate heat molding that can be used to make these kind of devices. And I will briefly describe it to you. So this is the fabrication scheme that I have shown you already. And what happens is at the beginning we create this mold. Once we have this mold can be used to fabricate this polydimethyl siloxane PDMS microfluidic devices as much as you want. You pour PDMS, you cure it, you remove it, you pour another batch of PDMS, you cure it, you remove it. You can use the same mold hundreds of times to make hundreds of microfluidic channels. But in order to make this full mold, you have to go to clean room, and it's a really hard one. It's like a pain in the ass to get a functional mold. And the thing is, the mold has a design on it, and sometimes design don't work. And you have to iterate to create the optimal design. And you have to go back to clean room, create a new mold, test it, and then change some design features and recreate it in the clean room. And you have to protect the mold that is finally working. Because if you break it, then you have to go back to clean room and make it again. And it is a really hard job, especially if you have a challenging geometry. So this is Bill Gates on top of the pile of papers when he first introduced compact disk. He is showing how much data that a compact disk can store in it. And this is me on top of the silicon wafers that I use to create one functional silicon wafer mold. That's why I say it's a time-consuming and hard job. So whenever you have this functional silicon wafer, you have to really protect it, although they are so sensitive and fragile. The other steps are easy, but for a project, you need hundreds of microchannels, right? So you really need to protect the silicon wafer. So these are all the microfluidic channels, or maybe some of them, that I made for this embryo compression project. These are the ones that failed. So I made a microfluidic cemetery out of it. So I asked if there's a way to copy these molds, so even the original master mold breaks, I can use the copy molds to make the same microfluidic channels. And this is out of serendipity I came up with this technique, and it worked fine. So you basically get the same mold. You made a microfluidic channel, PDMS, out of this mold, and then you use this channel and you put a polycarbonate sheet on it, and then since polycarbonate is a thermoplastic material, if you heat it up, that goes to the temperature that is beyond the glass transition temperature, it melts. It melts and covers this micro topology on top of the PDMS functional surface. And if you bake it for enough time, it completely covers this microsurface. And then if you cool it down, it solidifies back again. And if you remove PDMS, you create a mold that is exactly the same as a silicon wafer. These are just the side walls that defines the height of the PDMS, which is also useful. And you can use this polycarbonate mold to make the same devices here, the same PDMS, and you can use it. So I made tens of microfluidic channels using these copy molds, and I created copy molds for all sorts of different microtopologies. And here, let me show you some example. I made this micro pillar arrays that has only 2.5 micrometer in diameter. They look astonishing in my opinion. And I made this wavy surfaces, I made sharp features, I made semicircular microfluidic channels, I made multi-surface features, and they all worked. And I can unify all of them in a single polycarbonate sheet. And then I compare the dimensions of the devices that you make from original master mold and copied polycarbonate molds. And it looks like they can easily replicate the dimensions. So the dimensional fidelity of copying is really high. It's at least 95%. And they are, I tested them, they are functional as well. And then I want to test the limits of this copying. I got molds that has this diffraction gratings on it. These are the AFM images. And these waves are almost like 90 nanometers. So I went to the sub-micron level. And I copied them into polycarbonate and created this PDMS pieces, and I compared them. It looks like you can also copy these nanometer scale features, diffraction gradients, but the dimensional fidelity can be as low as 80%. So I wouldn't go down to sub-micron level, but it works really fine for micron-sized features. In summary, I created this molding technique called polycarbonate molding for copying the original silicon wafers, which enables us to multiply this silicon wafer molds, and since polycarbonate is tough, it doesn't break. Unlike silicon wafer, which is brittle, it's more reliable, I would say. And I tested the functionality and dimension, and I also showed that you can go all the way down to a couple of micrometers, but not lower than that. So this wraps up my last project during my PhD that I led. And now I'm going to San Diego to work with Ardem Patapoutian, who got the Nobel Prize in medicine this year. We are going to be working on mechanobiology using microfluidic devices. And he got his Nobel Prize by just working on mechanobiology. So that shows how important this field is. And if you like this kind of things, I would highly suggest you to this book that has just came out, which is about the history and the science and the technology of microfluidic systems, written by Albert Fork from University of Washington in Seattle. And this has just came out a couple of months ago. I just finished reading it. It's a beautiful book. And this book is dedicated to someone with whom we are familiar with. It's dedicated to Mehmet Toner, who is a graduate of this very faculty. He graduated from Istanbul Technical University Mechanical Engineering Department in 1983. And he is currently professor at Harvard Medical School, and he does amazing work using microfluidics. You definitely need to check him out if you are not aware of his work. And Albert Fork, who wrote this incredible book, is actually a student, a postdoc of Mehmet Toner. And Mehmet Toner is the guy who taught Albert Fork about microfluidics. And Mehmet Toner is currently one of the most cited microfluidics engineers in the world, and he's a leading expert in this field. And it came from our department. And which kind of implies, from the very conception of this field of microfluidics in 1964 by Cemal Eringen, who is a graduate of this faculty, all the way to 2024, Mehmet Toner, who is one of the most cited microfluidics engineers in the world, İTÜ Mechanical Engineering department, although indirectly, has a really big and crucial effect on this state-of-art technology of microfluidics. At the end, I would like to thank my advisor, which enabled this research. And I would like to thank my lab mates, but there are many of them, and I was too lazy to write their name one by one, so I drew them instead. And I would like to thank our collaborators, funding agents, and my space heater, which kept me warm in the never-ending cold days of Pittsburgh. Thank you for listening. That's it. Thanks. Sorry if I went too fast.
It's okay.
50 minutes.
I think we have some more time asking questions in Turkish or English, okay? Maybe we should start any question from the audiences physically from here. So var mı arkadaşlar? Yani yorum. Buyurun.
So, is the PC for the last project is the best option, first option? Is there any other thermoplastics?
There are definitely different thermoplastics. So the question that he asked is, why did you pick polycarbonate? Are there any other thermoplastics that you can use to do the same thing? The answer is yes, definitely. And there's a group right now in Carnegie Mellon who works on that, a new PhD student. And that's actually a future work that I'm really interested in seeing as well. And here you can see, let me show that to you. So there are lots of different material options that you can use. So they should be able to fit this process window. So this is the glass transition temperature of the specific thermoplastic material that you use. And this is the thermal degradation temperature of PDMS. If you go beyond 280 Celsius when you are heating up, the PDMS start degrading, so you lose all the microtopology. So as long as it fits to this process window, you can use different ones. Different material properties can affect that. There are too much detail and we can talk about that. But these materials and many others actually can be used. Why I used polycarbonate? That's what I had in the lab. There is no better answer. It's just that. There is no engineering work that I have done on different materials. I used polycarbonate because that's what I had, and I was satisfied with the results that I got. But there's a group that is working more on that.
So, to be able to achieve your good optimal results after that, you need clean room process?
No, definitely not. Once you have the silicon wafer, you can use PDMS parts that you make from silicon wafer. By the way, you asked, do you need...
The surface clean and keep
So he asked, do we need clean room to make PDMS parts out of molds? The answer is no.
But actually, it's not an issue. We have clean room storage.
So
not the mold, but to create PC mold.
you don't need it. So you can use an air gun to clean the surface, but what I do is I just use tape. So polycarbonate sheets are actually made in a very clean environment, and they have a protective layer on top of them. What you do is, right before processing, you remove the protective layer, and the surface is clean. But I use an air gun and also tape to remove any dust that may be on the surface, and I put it upside down and put it in a clean oven. And that works fine. I rarely have contamination on the surface, very rarely, almost never. So that worked fine. Thank you for the question.
Any other questions physical to
Show that you remove those pillars of PDMS out of wafers or
Mm-hmm.
PC molds. They seem like the aspect ratio is about 4 or something like that.
Exactly.
And did you do any further surface processing for removing that PDMS? Because as far as I experienced, I would break most of them.
Mm-hmm. Exactly, that's a huge concern, and one of our collaborators, so the question that he asked is, how do you make this high aspect ratio features? Why don't they break when you are removing them from the mold? And that's actually a huge concern, because if they break, then they remain inside the mold and you cannot remove them. It basically, although you don't break the mold, it renders mold unusable. So one of our collaborators asked me to use this technique to make copies of the original mold, and if it works, then if one of the molds that you multiply has this rupture issue, you can use the other one. But to minimize the rupture issue, which is very critical, we use silanizing agent. We coat the wafers with a silanizing agent, which decreases the stickiness of the surface. So the PDMS parts can easily be removed without much problem. It can still happen from time to time, but it's not super hard to get these. And I got them like for a really high surface area, like there are thousands of them in a very small area. They are really small, but it works fine. It works fine if you use silanizing agent to reduce the surface stickiness, I would say.
Another question from here, from a live institution. There's one question from Zoom. Dr. Rizade from Carnegie University. He's asking, how can you measure and control the shear stress in vitro, especially microscales?
Mm-hmm. So whenever we have, let me go back here. So I didn't again mention much about the characterization here, but I can talk more about it. Let me just go back here. So this is how you do it. Basically, even if you apply, so the question is how you measure and calculate the shear stress in this very small microfluidic channels. So as long as you accurately fabricate the microfluidic channels with the dimensions that you want to make them, then the flow inside these channels are laminar. They have low Reynolds number, all right? So they are viscosity-driven flows. The inertia can be negligible, which makes the calculations extremely easy because there is no turbulence. And there are really easy approximation of Navier-Stokes equations when you have this laminar flows. This is called Hell show flow. And you can, by just simple linear approximation, can calculate the shear stress that you create inside the channels and what should be the directions for a certain shear stress, or for a certain shear stress, how do you make the designs. You can easily do that. I can copy this to you and you can see it more in detail, but they're extremely easy because you have a laminar flow in short with low Reynolds number. So having small channels is not a disadvantage but advantage here, as long as you can fabricate them accurately, which we can do. Mm-hmm.
Because you need to measure the porosity of the channelizing to decide if it is laminar or turbulent.
Mm-hmm.
This is only just an assumption, right?
So we know the flow rates. We can create flow rates that we want to create, and analytically we can show that this is laminar. But I have experimentally showed this, which I think implies that the flow is laminar. I looked at the flow profile. So I used small microparticles, like 1 micrometer particles. I let them being dragged by the flow, and I measured their speeds at different heights of the microfluidic channel. And there's a parabolic flow profile, just like what I would expect from a laminar flow. So that experimentally, not perfectly, I haven't spent months on that, but I think it's fair to say that the flow is laminar.
Before concluding, I would like to get some of your ideas, specifically some suggestions to the young guys.
Mm-hmm.
Like there are so many PhD students, Bachelor of Science students, and also listening to you. What would you suggest them in terms of if they're gonna like to follow scientific career?
Mm-hmm.
Could you tell us something about that, based on your experiences?
Mm-hmm. First of all, you like to do what you are doing, and for me, the reason why I do is, the reason why I like to spend time in the lab is I have similar role models from my youth that showed me that science can be fun. One of them is Carl Sagan, the other one is Stephen Jay Gould. I really like Charles Darwin as well, and in Turkey, Celal Şengör kind of opened my eyes that science can be fun, and girls can like science as well. And if you study science, then you can have an internal motivation and you can get an external praise for that as well. So I do science and I spend lab just because of the selfish reasons. I know that technology can be useful or dangerous to the society depending on how they want to use it. So I'm not trying to help people, but I enjoy it. If you don't enjoy it, then go to industry and you will make a lot of money. But if you enjoy it, then you will be part of a 2,500 years old academia, which is a really still intellectual organization, the best intellectual organization in the world, I will say. And being part of it makes me feel good. How you can start doing that? Maybe you can start following what the professors that you have in the department are doing. I started joining the labs in the lab of Emin Sümbüloğlu after I took strength of materials class from him, and then I joined Dr. Trabzon's class, and I took part in his lab. So if you do these kind of things, then you will enjoy. But maybe you can start by reading some popular science book, and maybe you can see if you're going to enjoy them or not, especially if you don't have any academics in your family. That's the first step that I can say. The rest will be easy because you are studying in a really hardcore university. Maybe they don't praise you, maybe it's hard to get good notes, but no matter what, if you spend a little bit of an effort, you will see your friends that may be coming from Harvard, MIT, all the great technical universities are not better than you. The education, the undergrad education that you get here is amazing. That's what I have seen. As long as you motivate yourself, you start the game from far beyond compared to other people. And the history of the university shows that too. I'm not saying the same thing for research. Research in Turkey is tricky, but the undergrad education here that you get is, you're well lucky, and I was lucky, that is amazing.
Thank you very much. If not any other questions, let's thank him one more time. And we will now conclude the talk. Thank you very much.
Thank you for listening.